All right, welcome back to the next to last lecture. This is the biggest one. The next one's just kind of um, a short lecture related to these enzymes. But this is the big one, enzymes. This is the big one. And so let me go ahead and show you our objective here. Welcome back, by the way. So I'm going to use this board in a way that you can see it. So let me turn around because I think this camera angle is not going to show it. All right, there you go. 16.9, let me make sure my microphone is close enough. Okay, so 16.9 is about enzymes. Now, enzymes are key for a lot of things that happen in our body, and that's one of the big jobs of proteins. Well, I don't know. Proteins are really good for structure. If you don't have structure, nothing's going to happen. So proteins are great for structure, but kind of the fun stuff is what enzymes can do, or what proteins can do when they act as enzymes. So the first thing we'll discuss is what the characteristics of enzymes are. Like, what are their properties, and just a little bit about their nomenclature. We're not going to go into a huge nomenclature system of how enzyme nomenclature works, but it actually is pretty simple anyway, and so you'll see how simple that is. But at least we want to cover that so that we can start having the conversation of how they work. And there's two different ways that they work. There's the lock and key type model, and there's an induced fit model. And then finally, we'll spend a little bit of time talking about inhibitors and how those work on stopping enzymes from doing their work. And there's two different ways that inhibitors can work. They can be competitive, or they can be non-competitive with the substrates that the enzymes are acting on. So most of our conversation is probably going to be in B on how they work, but we really need to spend some good time right now talking about A, their characteristics, and a little bit of their nomenclature, because we're going to be using the names of enzymes a lot. And so let's go ahead and get into this. Let me set this down. All right, and then let me jump to the PowerPoint here. All right, let me minimize that. And boom, here we are. Let's scroll up to 16.9. Here we are, enzymes. So basically, they're going to be proteins. That's why they're in the chapter proteins. And they're going to serve as biological catalysts for reactions in all living organisms. That word catalyst means to speed up or to help occur. So oftentimes, I need a catalyst to make things happen, like an alarm clock to get me out of bed, or a little bit of and never mind with the analogies. Catalysts are needed to make things occur. They're not usually the thing that does the stuff, but they are what's needed to make the things that are going to do the stuff, if you know what I mean. So I'm speaking in very vague terms, just so you have a good idea of what catalysts are. Catalysts are necessary for either making the chemical reaction occur altogether or simply just speeding it up. And so, like all catalysts, enzymes are going to increase the rate of reactions by speeding them up. But they themselves are not permanently changed in the process. So that's important to memorize. Important concept about catalysts. They're not like literally reactants. Like if you look at a chemical reaction here. See my mouse pointer here? If you look at a chemical reaction here, you'll see the enzyme or the catalyst underneath the arrow or above the arrow, but that's not necessarily a reactant. But it is needed, right? It is needed. Like it's usually an acid or a base, or in biological mechanisms, it's going to be some molecule like catalase or carboxypeptidase. And so you'll see ACE a lot, and we'll talk about how that happens. And so enzymes are crucial for allowing biological reactions to occur. And most of the time, enzymes must work under certain conditions. Like they have to usually work around a pH of 7.4, which is the pH of your blood, 37 degrees Celsius, which is the temperature of our body. And so it's very important that we keep those pHs and temperatures in our body so that enzymes can work. And so what are the characteristics of enzymes? Well, basically, they're going to also obviously be water-soluble globular proteins because they're moving around in our blood. So they need to be water-soluble. So they're going to have a lot of hydrophilic side chains on the outside and hydrophobic side chains towards the middle of the globular protein. And they're also going to be very specific in what they do. So some of their characteristics are that they greatly enhance reaction rates. So they speed up chemical reactions, and they're very specific. Now, the specificity of an enzyme does vary. Some enzymes, such as catalase, for example, catalyze a single reaction. Like catalyzes the conversion of hydrogen peroxide, H2O2, to O2 and H2O. So if you put hydrogen peroxide in a bloody wound or whatever, and you want to clean it, basically you see all those bubbles, that's what's happening. You're making the oxygen bubbles. So the hydrogen peroxide is reacting with the bacteria and this enzyme catalase to make O2 and H2O. So if you didn't have catalase, it wouldn't happen. So catalase is very important for just that. Other enzymes, such as carboxypeptidase A, they will catalyze a particular type of reaction. And in fact, it's in the name, which we'll talk about in just a second. It's always going to be some carboxypeptidase. So peptide is breaking the peptide bond near the carboxy end or the C-terminal end of a peptide. So if you see here, you have the N-terminal of a peptide and the C-terminal end of a peptide. Well, carboxypeptidase A is an enzyme that usually breaks this peptide bond, but not this one. It's breaking this one because it's closer to the carboxy end, right? It's closer to the C-terminal end of this peptide. And so it's not going to break this one, right? And that's, but it could do that on a variety of polypeptides. And so, but in general, it does the same type of reaction, but it's not as specific as catalase, where it literally is just always doing H2O2 to turn it into H2O and O2. So there's a little bit of variation in specificity, but nonetheless, enzymes, by looking at their name, are very specific. And that's what's cool about enzymes. We don't have to learn this like huge nomenclature system necessarily, but a little bit about it is that the names are always going to end in the suffix ase. So if you see ase, then you know you're dealing with an enzyme. And then whatever comes before that is basically the type of molecules that it's working on. Now, the type of molecules that an enzyme works on are called substrates, by the way. So we haven't got to that word yet, but basically the substrates that the enzymes work on are going to be part of their name. So for example, an enzyme that catalyzes a hydrolysis reaction is called hydrolase, right? Now it's not water. Water is what does the hydrolysis, but hydrolase is what speeds that process up. And hydrolases was just kind of a generic vague term, right, for any enzyme that can speed up hydrolysis. But you can hydrolyze proteins like we were talking about in this chapter, in the last lecture, in fact, or you can hydrolyze lipids, right? And so you could further subdivide hydrolases into lipases, which will hydrolyze lipids, or proteases, which hydrolyze proteins, right? Proteases are going to catalyze the hydrolysis of proteins, and lipases are going to catalyze the hydrolysis of the ester bonds, if you don't remember, in lipids. So if you're going to hydrolyze lipids, you're breaking those ester bonds. Okay, here's an interesting reaction, by the way. The conversion of lactate, that's the same thing as lactic acid. Anybody ever get lactic acid build up in your muscles and you get sore? It's lactic acid. So the conversion of lactate to pyruvate is a very, very important feature of an enzyme catalyzed reaction, and very important in our body. Otherwise, if you had lactic acid build up in your muscles, it would always be there and you would be sore all the time. And so how would you ever get rid of the lactate? You have to be able to get rid of it. And so here's how. You're gonna use this enzyme, this catalyst called lactate dehydrogenase. And so again, the name tells the story. It's acting on lactate, so that's why it's called lactate dehydrogenase. And the process, the type of chemical reaction, is a dehydration reaction. Dehydration reaction or any dehydrogenase is going to be something that catalyzes the removal of 2 hydrogen atoms from a substrate. So our substrate, remember, is always what the enzyme is acting on, or the catalyst is acting on. In this case, lactate dehydrogenase is acting on the lactate, and it's doing a dehydrogenation. It's doing a dehydrogenation. It's taking away 2 hydrogens. You can see them here in red, one there and one there. And so this is lactate or lactic acid. And basically we're taking these 2 hydrogens and we're going to put them in 2 different places. One of them is going to be put onto coenzyme. So oftentimes you need more than one enzyme. And this enzyme here, coenzyme NAD+, is actually what's going to be really participating in the chemical reaction. It's going to gain one of those hydrogens and become NADH. So we're going to go from NAD+ from NADH. And then the other hydrogen is just going to be a free-floating proton like that, just a hydrogen ion. All right, so that's how we're able to take lactate and get rid of it. Our body's going to turn it into pyruvate. Now this is interesting. If you remember, pyruvate then can go through the Krebs cycle and the cellular respiration and the electron transport. Krebs cycle and electron transport chain are in the mitochondria and make up the final process of cellular respiration. And so just for fun, I thought I'd take you guys to this picture here. This is a cell. Here's a cell membrane. Some glucose coming into the cell, and you guys remember the main part, the first part, glycolysis, because of that song probably. Glycolysis, come on sugar for the breakdown. And then about the pyruvate, the final destination of pyruvate is going to depend on what happens to pyruvate in the end. Whether it's fermented, if there's oxygen, it'll go through this process. If there's no oxygen, then it'll be fermented. What's cool is you can still make ATP through glycolysis. Most of the ATP is made in the mitochondria through the electron transport chain. But you can still make ATP through glycolysis. Remember, ATP is like a charged battery. And so ATP is what we use, is the fuel that our cells use for energy. And so we don't use glucose for energy. We break it down to make ATP. Right? ATP is the currency in our body. It's like the $1 bill. But glucose is like gold because it can be sold for a lot of money. A lot of ATP is the real currency. All right. And so basically, again, if you have oxygen, then after glycolysis, pyruvate will go through the Krebs cycle, the citric acid cycle, and the electron transport chain and make a ton of ATP. And so if there's no oxygen available because you're either lifting really, really heavy weights and you just can't meet the demands of oxygen for all the mitochondria in your muscles in that millisecond, or you're taking off from a race and you need a bunch of energy to start the race, or you're further into the race and you can't breathe fast enough to start to go into the anaerobic phase. That's when you're going to be sore the next day, by the way, because you're going to be doing lactic acid fermentation. Now, here's what's cool about lactic acid fermentation is you're going to be making lactate in order to do this, in order to take nicotinamide adenine dinucleotide and turn it into NAD+, nicotinamide adenine dinucleotide without the hydrogen. And it's these right here, the NAD pluses, that you want, because those are necessary to power glycolysis. You can have all the sugar in the world in your body, but you won't do glycolysis if you run out of NAD pluses. So you need to replenish those NAD pluses in order for this chemical reaction to occur. And so fermentation is just the way that happens. Your body doesn't do fermentation to make lactic acid, because we don't want lactic acid, but that's a byproduct of pyruvate. And you're doing that so that you can make NAD plus to keep powering glycolysis. Well, now check this out. The reaction we just looked at is the opposite. If you want to get rid of lactate, you use NAD pluses that are available, and you use lactate, and you turn it into pyruvate. So it's the exact opposite reaction. And so my question is then, if you have lactic acid, then can't you make pyruvate and then let that get turned into ATP? Sure. You sure can. So it's going to need pretty little cyclical process there. The lactic acid you have can literally be used as energy later, but in a different way. So it's better to just do glycolysis, just to metabolize glucose through glycolysis and take it through the mitochondria. So pace yourself when you're running so that you got plenty of oxygen and you're always in the aerobic phase. If you're in anaerobic phase, it's not necessarily bad, but it's not good for energy's sake. Okay, now the reason I brought that up is because there's another illustration in the lecture that I want to show you guys about another enzyme that has to do with the same process. And so the enzyme for this process to occur is called lactate dehydrogenase. There you go. It's a name derived from the substrate, which is lactate, and the type of reaction, which is dehydrogenation. That is the removal of 2 hydrogen atoms. And to carry that out, a cofactor is needed. A cofactor is the same thing as a coenzyme here. A cofactor is essentially it could be 2 things. It can be a metal ion or just some non-protein organic molecule, but not a protein. Any kind of non-protein organic molecule or a metal ion that's needed for an enzyme catalyzed reaction to occur is called a cofactor or coenzyme. So it's like, man, you need these enzymes that are acting as catalysts, and then you also need another enzyme, a coenzyme, to work. And the coenzyme is usually what does the work, like the reaction part. Like in this case, NAD+ is participating in the chemical reaction with lactate. Lactate dehydrogenase is just the catalyst. But both of these are considered enzymes because they're proteins. I'm sorry, no, this one is the protein. This one is a protein. This is a coenzyme, but it's not a protein. That's why we call it co. It's like a co-pilot. Not really a pilot yet, but so NAD+ again is the nicotinamide, it's hard to say, nicotinamide adenine dinucleotide, and it's the cofactor that oxidizes lactate to pyruvate. So I think we can move on further now of how enzymes work. So basically, this is really cool. This is basically how like all enzymes work, and it's so simple to understand, especially when we're looking at this visual here. I'm a very visual person, so I get excited when I see this kind of stuff. An enzyme contains a region. So here's our big protein, and it's an enzyme because it's doing a job, speeding up a chemical reaction, and it's acting on a substrate. Here's our substrate. And the way it works, at least in the lock and key model that we'll discuss first, the way it works is you have this active site. It's almost like a landing pad on the enzyme, and the substrate fits like a lock and key. See how it fits like a lock and a key? The substrate's like the key that fits into the lock, the enzyme. And then the enzyme can do its job, whether that's breaking these bonds and then releasing the products, or there's variation to what sometimes it literally just gives the enzyme a different shape and then will allow the enzyme to do something then. Or it'll allow the opposite to occur. Like you see here, you're taking a substrate and breaking it up into 2 different products. Sometimes enzymes work by taking a substrate and then taking another substrate and then putting those 2 things together and then releasing the thing as one, right? Almost like like a pastor would take a man and woman and marry them and send them off as I now pronounce you husband and wife, right? In this case, this would be like a divorce attorney saying, okay, now you're separate. But you needed the enzyme, you needed the divorce attorney to do that. They couldn't just say, hey, I don't want to be together anymore. All right, sorry to use a kind of a weird analogy there. So that's the lock and key model. The other is the induced fit model. So this one's not quite like a lock and key, but it's a little bit different. It's because, again, in the lock and key model, the shape of the active site is rigid. So this is a rigid shape. So like no matter what, it's always going to act on this specific substrate or whatever fits in there, okay? But the induced fit model is a little more flexible because what happens is it's thought that when the substrate and the enzyme interact, the shape of the active site can adjust to fit the shape of the substrate. that make sense? It's thought that when the substrate and the enzyme interact, the shape of the active site then can adjust to fit the shape of the substrate. So it's kind of like, oh, hey, you're here? Okay, let me make room for you. Let me clean the couch off and let you sit down. So these are a little more flexible. That's just a different type of model. But basically, it still works almost like a lock and key still, right? This shape fits this shape. So pretty simple concept. I love it. It makes understanding this so easy. Now, of course, there's just thousands and thousands of substrates and thousands of enzymes that do different things, and so that's where it gets hairy in biochemistry. But once you have this down, you can start having those conversations and studying the stuff with peace and confidence. So this model is often used to explain why this one right here, this induced fit model, is used to help explain why some enzymes catalyze reactions in a wider variety of substrates. Like the scientists have noticed, hey, this enzyme works on not only this substrate, but this substrate too. And so it's because the shape of the active site and the substrate must be reasonably similar, but once bound, the shape of the active site is different from the shape of the active site in an unbound enzyme. And here's the example they use that I wanted to bring us back to that cellular respiration image that we were looking at. So here's an example of hexokinase doing its job via the induced fit model. So here's hexokinase. You can just look at that word, hexokinase. Obviously that's working on a hexo sugar, and so here it is, glucose. The way it works is here's the active site in the middle, and when glucose gets in there, the shape of this enzyme will change and almost bury the glucose inside. And then once that's done, hexokinase can do what it does. Now let me show you what hexokinase does. This is kind of neat. Hexokinase, remember we said that glucose can go through cellular respiration and it requires a little bit of ATP. Well, what is ATP? If you remember, ATP is like a charged battery. It's adenosine triphosphate. So it's adenosine, anaribose, and 3 phosphates. The 3 phosphates, especially the third one, is what makes it a charged battery. If you rip off that phosphate, and you have 2 phosphates, then you have adenosine diphosphate. Di means 2. And so that would be like a dead battery or a low charged battery, because there's energy stored in that bond that holds that third phosphate on. And so, but still, what do you do with that phosphate? You still got to do something with it, right? You got to take this phosphate and put it somewhere if you're going to use up the energy. And so that's what happens. The cell takes ATP and it utilizes that charged battery and it plucks off that phosphate. And then what it does is it puts it onto glucose to make glucose 6-phosphate. It just jams a phosphate onto glucose. So you go from you lose a phosphate. That's why you go from ATP to ADP. Now you need hexokinase to do that, and then you have glucose 6-phosphate, which then can go through glycolysis, hooray, energy, or you can use that molecule as in an anabolic process instead of a catabolic breaking down process. You use it in an anabolic building process to make other molecules, or you can make glycogen or starch. Again, big polymers, large polymers of polysaccharides is what we call those. Remember, it's glycogen and starch. Glycogen in your muscles for storing glucose, starch in plants. Okay, so pretty cool, huh? Hexokinase does that, but hexokinase is one of those that is like an induced fit sort of thing. Like it gets in here and then it changes its shape. And my hypothesis is, and you can correct me if I'm wrong, is that once it changes shape, that's when it can grab the phosphate and jam the phosphate onto the glucose. It's like a pretty fluid little process there. And so correct me if I'm wrong. The book didn't say it, but I think that's what happens. Okay, now let's look at inhibitors. That word inhibitor means to slow down or to stop the activity. So an inhibitor is a molecule that causes an enzyme to lose its activity. So how do you think that works? Who knew? By changing the shape, right? By affecting the shape. So an inhibitor, all this stuff is based on shape. The way an inhibitor works is by reacting with the enzyme and sticking to it. through a chemical reaction. And we'll look at this one in just a second. But in general sense, an inhibitor is a molecule that causes an enzyme to lose activity. So there's all kinds of inhibitors that you can take as pharmaceuticals, or your body naturally has them, and they slow down chemical reactions. They're like the opposite of a catalyst. An inhibitor can do their job in two different ways. They can either do it in a competitive way or a non-competitive way is the main thing that we're going to discuss. But first of all, they can do it in a reversible way or an irreversible way. And so a reversible inhibitor binds to an enzyme, but then the enzyme activity is restored once the inhibitor is released. So if the inhibitor doesn't stay there forever, and can be taken away, and the enzyme can go back to doing its job, then that's a reversible inhibitor. But an irreversible inhibitor is one that literally covalently binds to an enzyme, and that's permanently going to destroy its activity because it's permanently going to be there. It's going to be a permanent covalent bond. And so penicillin is a great example of the way that works. And so penicillin is awesome. Penicillin, the way it works, it's an antibiotic that kills bacteria by preventing it from making its cell walls. And the way it prevents the bacteria from making their cell walls is that it irreversibly binds to glycopeptide transpeptidase. Okay, again, you can analyze that word, and it ends in ase, so we know that is a enzyme or catalyst. It's an enzyme, and that enzyme is acting on glyco, that means sugar, peptide, that means a carbon-nitrogen bond, remember, a peptide bond, and then transpeptidase. Anyways, we're not going to do that for this enzyme here, but again, you can analyze these names to figure out what the enzyme's doing. But they tell you right here, it's an enzyme that's required for the synthesis of a bacterial cell wall. And so if penicillin is going to come around and bind to this hydroxyl group or this OH group or this alcohol functional group that's on this enzyme, then it's going to inactivate it, and it's going to halt the cell wall construction, which is end up going to be killing the bacteria. So isn't that cool? Here's the penicillin. Here's the penicillin molecule. I think one of you guys, Peter, did this for his organic chemistry presentation. And the way it works is, I look, you can see it right here and here. The way it works is this oxygen, this bond right here is broken. You see how you have a square? You don't have a square here. That square is opened up. So this bond right here is opened up. And it's a peptide bond, carbon-nitrogen. It's a peptide bond. And so the way that works is this carbon now is going to bond to, instead of this nitrogen, it's going to bond to that oxygen. And this hydrogen is going to bond to that nitrogen to fulfill that guy. And then you end up with this situation here. And that's going to be a covalent bond there. So that covalent bond there, which again is not an ester, then that's going to be keeping the penicillin on that enzyme, rendering it inactive. And so that's a pretty awesome thing. Now, unless that's not what you want, then that could be a bad thing. But so reversible inhibition, the reversible kind, going back to the reversible kind, they can be competitive or non-competitive. So, for example, here's a good picture. Let's start with a non-competitive. A non-competitive inhibitor binds to the enzyme but doesn't bind to the active site. So it's not competing with the substrate. So what happens is you have this active site, the substrate will normally fit there, right? Well, if you have a non-competitive inhibitor, it's not going to go there. It's going to go somewhere else on the enzyme. And when it does that, it changes the shape. It's like a bully that comes around here and pushes on the enzyme and makes these guys move and kind of messes up the structure. So now the substrate no longer fits. If it no longer fits, the enzyme can't do its job, right? So, but that's not really competing with the substrate. Okay, another model is the competition model, where literally I wish they had a visual. I can't believe they don't. Or do they? No, they don't. Wow, interesting. A competitive inhibitor would be one that looks just like the substrate and fits at the active site so that the substrate can't fit there. It's like the bully that comes and literally takes your seat and doesn't let you sit down. And so they don't have a nice picture like that, but they do have an example here. So this is a really cool situation. So follow this train of thought. Here's a competitive inhibitor called sulfanilamide, and it's an antibiotic. So if you ever get sick and you got to take an antibiotic, here's one of your options. Sulfanilamide. When your doctor gives it to you, you can tell them you know how it works, and they'll be like super impressed. The sulfanilamide, it's a competitive, remember, inhibitor, and the way it works is it literally directly competes with this molecule here, amino benzoic acid. You see how they look real similar? They look very similar, right? So the antibiotic sulfonamide, it's a competitive inhibitor of the enzyme needed to synthesize the vitamin folic acid from the amino benzoic acid. All right, in bacteria. So bacteria are normally taking this amino benzoic acid. We'll just call it PABA. It's normally taking PABA and turning it into folic acid, right? Which it looks like it's happening right here. Like here's the amino benzoic acid, and it would normally go right here. But, and of course other things are getting added on too, but the point is, if you don't put this there, then you won't get folic acid. If you put this there instead, sulfanilamide, you won't get folic acid. So you have to have amino benzoic acid to come together with these other molecules here to make folic acid, right? You see the double bonded oxygen there, double bonded oxygen there. You can't have this sulfur and all this junk. That's not going to work, because then you won't get this guy to bond there. You'll probably get the hydrogen you'll probably get that bond there maybe. In fact, that's probably exactly what will happen. So isn't that pretty neat? And what's really cool, check out this last sentence. Sulfonamide does not affect human cells because humans do not synthesize folic acid. We eat it. That's a vitamin that we eat. It's one of those essential vitamins that we have to eat because our bodies don't synthesize it. Remember, the vitamins are like the lipids, and so we synthesize some of them, but some of them we don't. All right, cool. So there you go. The next lesson we'll look at is just real short, talking about how enzymes, we're focused on health again. We'll talk about specific enzymes and how they can be used as diagnostic tools, and then that's it. All right, so I'll do a short little video on that lecture, but this one was kind of long, okay, because there's a lot going on here with enzymes, and those examples were really cool. There's thousands of enzymes out there, but I will hold you accountable on the test, which would be open book, by the way, so you at least have a resource to look some of this stuff up. Now, there are going to be a few things, though, that I want to include that might not be in the book, so keep your eye out for that. And one of them, let me just go ahead and announce it now, but I'll make a Google Classroom. Let me cut to my different camera angle here. I'll make a Google Classroom assignment for it. That is, what I want you guys to do is I want you to consider all of this that you've learned about proteins. All that you've learned about proteins. And hopefully it has to do with enzymes and substrates and the whole lock and key model, but I think I'm going to have to show you this real quick for the assignment. So going back to the lecture here, what I want you to do as an assignment, a 1 page to or half a page to a page, however long it takes. I'm not looking for length. I'm just looking for a good explanation. You see this? I don't know if you guys got this. I might have to send it to you in another PowerPoint or whatever, and I'll give you other pictures to look at too. This is the coronavirus, and this is the way the coronavirus works. The way it works is that there's proteins. These proteins on the outside of the coronavirus are kind of all the buzz is about the coronavirus. It makes them unique. So you've probably seen the images of the coronavirus and all these little spike proteins. Well, there's all kinds of proteins on the outside, but it's specifically that spike protein that we're worried about because it's it's like the mutation part of the virus. It's the new protein that's there. And the way it works is these proteins react with these receptors here on your lung cells. And once this reaction here occurs, like the lock and key, then the lung cell will engulf the coronavirus. Well, once it does that, you have a bunch of RNA inside of the virus, which is the next chapter, by the way. And so hopefully if you were pay attention in biology in freshman year, you should have an easy time understanding DNA and RNA and the way it works. It literally codes for making proteins. The chapter we're on right now, polypeptides and proteins, the amino acid sequences. Amino acid sequences are dictated by nucleotide sequences in RNA. And so that RNA will get translated. Here's a section of RNA. They'll get translated into this protein. This section will get translated into that protein, and this section into that protein. And all those proteins will get attached onto some rough endoplasmic reticulum that'll bud off and go to the Golgi and be packaged and turned into another coronavirus with all those proteins on the outside. So if we can, anyways, I'm not going to go into that tangent. What I want you to focus on for that assignment is, and I'll get more into details when I make the Google Classroom assignment, is I want you to focus on this mechanism right here, this attachment mechanism. What's happening here with this spike protein? I want you to do your research. This is stuff I don't know yet, by the way. So I'm wanting to know, I'm wanting to learn, and I'm wanting you guys to learn. Do as much independent research as you can and figure out what is going on here with this protein, this spike protein, and this receptor. What kind of mechanisms are occurring? If you can find some good images to also include in your assignment, that'd be awesome. And just some short descriptions of what's happening there so that we can better understand that. Because really, at the end of the day, if we can prevent that process from happening, then the coronavirus is not getting into our lung cells, right? And in fact, that's how some of the drugs are working, is they're attaching to the spike proteins or attaching to the receptors in a competitive way so that the spike proteins can't fit there. It's a competitive process. Problem is those receptors are originally designed for something else, and you might interfere with that. And so I don't know about all that. That's why I'm asking you to do some research. Anything you can about the coronavirus and how those proteins enable the recognition, even if it has to do with the history of how that protein has changed, because technically the coronavirus is just your common cold. In fact, it's less severe than the flu. Influenza is a different type of virus. The flu virus, influenza, is very different than the coronavirus. The coronavirus is a family of viruses that look like the coronavirus, but the proteins are different, and that spike protein is what's different and is mutated into such a way that the coronavirus is recognizable by humans. And so if you can figure out what that protein is that got mutated, or whatever it is, it would be awesome information to see or read about. And so thanks, guys.